Conversations That Matter is a partner program of the Center for Dialogue at Simon Fraser University. The production of this program is made possible thanks to the support of the following and viewers like you. Welcome. I find this to be kind of an exciting time for people who are dealing with diabetes in general, but in particular type 1 diabetes. Just for anybody who might be interested, give us a brief description of what type 1 diabetes is and how it differs from type 2.
Right. So type 1 diabetes is the form of diabetes in which the patients can no longer produce insulin. That's in contrast to type 2, where at least initially, patients with type 2 diabetes produce the insulin, but their bodies don't respond to the insulin. So in type 1 diabetes, what has happened is that the patient's immune system has sort of backfired. We call that autoimmunity. And it's killed the cells in their pancreas that are called beta cells that make the insulin that allow them to regulate their blood sugars.
Do we know yet why the body does that in some people, why it will kill those beta cells?
Why that autoimmunity develops in the first place is an intense area of investigation right now. There are some clues. There is a genetic component that is well described, but we're now finding there's likely also an environmental component to what triggers that autoimmune reaction.
I've heard it described as being told, well, there was a car accident at this intersection. There were cars involved, people were hurt, somebody died, but we don't have the cars, we don't have the people, we don't have the witnesses. Now figure it out.
Yeah, that's a really great analogy, actually.
Yeah. And so that makes it all that more difficult.
Yes.
So we go back almost 100 years ago now to Banting and Best and their discovery of the benefits of administering insulin. Then we went this long stretch with very little developments. There's been some exciting things happening over the last couple of decades as far as the treatment of type 1 diabetes. But I think most importantly for people, when they take a look at the Edmonton protocol, they started to think, oh, okay, maybe there might be a cure. But there's challenges there. What are some of those challenges when we take a look at the transplant option that comes through the Edmonton protocol?
Right, so transplantation for type 1 diabetes has made sense right from the get-go. You have a very specific cell type that is lost. Ideally, you'd want to replace that cell type. That's the beta cell. And that has taken the forms of whole pancreas transplantation and, as you allude to, islet cell transplantation, which is really innovated on the front of immunosuppression therapies, so improved immunosuppression, originally described as the Edmonton protocol, piloted by James Shapiro and his colleagues up at University of Alberta. And the problem, now that they've enhanced the immune suppression, is the cell source. There aren't enough cells to treat how many patients you would love to treat with type 1 diabetes, so there's only about 1,400 pancreases available each year in the US, at least. I'm familiar with those numbers. And we have over 1 million people with type 1 diabetes, so it's easy math that you need a new source for replacement islet cells and beta cells.
And so where are we at right now? What's our next step? What's the solution to that?
We believe the solution is stem cells, and in fact pluripotent stem cells specifically, because they have an immense proliferative potential. So each patient might require on the order of 300 million cells to be delivered as their therapy. And you really need a stem cell that has great proliferative power to make that many cells and to treat that many patients. So that's where pluripotent stem cells come in because of their proliferative potential. But the challenge with that cell type is harnessing its cell fate, right? So we don't want to deliver pluripotent stem cells to any patients. We need to deliver islet cells or at least pancreatic cells. So the bottleneck in the field for over a decade really was learning how to take that stem cell and change it into pancreatic cells. We call that differentiation, or the process of differentiation, where the immature cells become more mature cells. And that's a big part of where our company, ViaCyte, is focused through the years, is on harnessing stem cell differentiation potential and coming up with a recipe for changing those stem cells into pancreatic cells.
So what was the process that you went through to be able to do that? because I'm sure that it's not, you can describe it in a minute or two, but the process of figuring out how to make that work has to have been challenging.
It was very challenging. We had a team of over a dozen PhD scientists working iteratively on this process for almost a decade, to be truthful. So beginning around 2002 and up until probably 2012, literally 10 years, that process was in constant flux. Now, since then, we've been able to lock in our process for doing that, do all the safety testing and the quality testing that's needed to move towards clinical application of that. And that's where we are today.
So I got to get you to just hang on for 1 second while we take a quick commercial break.
Okay.
We'll be right back. Conversations That Matter is a not-for-profit program made possible thanks to the charitable support of the following and from viewers like you. Please visit conversationsthatmatter.tv and help us to continue to produce this program. Thank you. So what were the biggest challenges in that developmental process to be able to get to a point where you can deliver something that is now at that human trial stage?
Right. The biggest challenges, there had been researchers studying the developmental biology of the pancreas and the islet cells in animal models and even in humans. And they provided the framework that we were trying to follow in the lab, in the Petri dish. However, we don't have organ boundaries. We're not forming an actual organ. So they study how an organ forms, and we're trying to study and replicate the formation of cells in the absence of the organ. So it made it more complex.
And those cells within the organ.
Yes, very specific types of cells within the organ. So we wanted to make not the whole pancreas, just the islet cell types. Mm-hmm. And islets are only about 2 to 3% of the total cell types in the pancreas itself. Most of them make digestive enzymes, and we're interested in the ones that make the hormones.
That respond to the K cells that, as I understand, deliver the signature, I guess, that comes from elevated blood sugar levels back to the pancreas and says, okay, now produce insulin. So it's not only are you producing the cells, you got to make them communicate with the host body.
Yeah, and that's where I think the biological approach here of cell replacement, the advantage is that the biology works, right? We needed to know or figure out a way to replicate that biology in the lab and harness that as a mode of therapy. So that's where cell replacement and cell therapy for diabetes is really makes a lot of sense, for type 1 diabetes especially.
So if it was an autoimmune disease in the first place that destroyed those cells and you're now replacing them, are those new cells not also susceptible to that same autoimmune disease going back to work again?
Yeah, they absolutely would be. In fact, they'd be susceptible to rejection because the cells aren't matched to the patient.
Right. So there's also the rejection component.
Right,
Yeah.
and they'd be susceptible to both the alloimmune and the autoimmune rejection processes. And that's why we're developing a combination product. So it's not just a cell replacement therapy. It also has an encapsulation device component to it. And that encapsulation device is meant to protect the cells from both the alloimmunity, because it's not matched to the patient, as well as the patient's own autoimmunity. And it does so by eliminating contact of the patient's immune cells with the cellular graft.
Because it's within a protective coating.
Exactly. It's essentially it's a medical device. It's got a very thin membrane, and the purpose of that membrane is to eliminate cell-cell contact. And in doing so, it eliminates the effector mechanisms of the immune system so that the cells cannot be rejected. It's almost, you can think of it like a tea bag. It's a pretty fair analogy where
It's
the
porous.
cells, it's porous, right?
Yeah.
The tea is like the cells, so things can pass through freely, but cells cannot get in, and also cells cannot get out. So the advantage of this encapsulation approach that we've developed actually has safety advantages as well, and it's what allowed us to get into a clinical trial relatively rapidly because of the safety advantages provided by the device, and that the cells cannot escape. They can't go anywhere, so we can put cells in and we can take every cell back out by surgically explanting that product.
So tell me about the clinical trials. What was the the model that you employed, and what were the results?
So the clinical trial initiated last fall in September 2014, and we're still ongoing in that clinical trial. It's a phase 1/2 trial that's meant to evaluate the safety and tolerability of the product first and foremost. And once we get to the second cohort of that trial, it would also be meant to evaluate early efficacy of the product. Right now, we're still in the first cohort of that trial. We've implanted 10 patients so far, and we're learning a lot. You know, it's an open label trial, and we're working with both University of California, San Diego, researchers there, which is really right across the street from us, as well as Dr. James Shapiro up here at University of Alberta at Edmonton. And as we discussed, he was really an innovator of islet transplant, so it's wonderful to have him participating in this trial now.
So what's the most encouraging results that you're seeing right now? I know that you have to say, I can't tell you all these things and nothing's definite until it's done, but you must be getting some early results that are encouraging. And I ask because people who are dealing with type 1 diabetes are keen to know.
Right, they are, and it is encouraging. You know, no one has ever done anything quite like this before. A combination product like this, a stem cell derived product that is both encapsulated, it's a combination product, it has sort of a fairly complex regulatory definition there. And it also requires surgical implant and explant that no one had ever done before in people. We had done it in animal models and pigs, but you know, humans are not mice, rats, and pigs, of course. So there have been early learnings there on how you do the surgical implant and the explant most expediently, minimizing any pain to the patient, and we've made great strides there, and it's been very well tolerated. So,
got to take another break. We'll be back in just a moment. Conversations That Matter is a not-for-profit program made possible thanks to the charitable support of the following and from viewers like you. Please visit conversationsthatmatter.tv and help us to continue to produce this program. Thank you. Well, considering where the pancreas is buried in the core of your body, getting there is not easy. How do you get there? Do you go in here or in through here?
Well, that's the interesting part, is we're not going into the pancreas at all. So
You're not.
This is an implant under the skin. We call that subcutaneously. And the reason we can do that is what we're replacing is the endocrine function of the pancreas, so the hormones.
Yeah.
And the definition of endocrine cells and hormones are that they act systemically. So all that needs to happen is the cells inside of this product need to have access to the bloodstream, so to speak. You know, they need to be able to sense the elevated blood sugars. When they do, they would release insulin that then goes back across that into the blood and circulates throughout the body.
So the insulin won't be coming from the pancreas?
The insulin does not come from the pancreas. It would come from the product implanted under the skin, wherever that would happen to be. We've also been evaluating anatomical sites for where those implants would be most conducive to both patient comfort, and ultimately, once we get to second cohort, we could evaluate efficacy among those as well.
Wow. My mind is racing at the moment thinking about what it takes to do that, because you have to be glucometer and injection site at the same time, and it has to happen without any human interaction.
Yeah.
I'm right about that.
Yeah, well, this is the biology. So the beta cell is, you know, it's exquisitely good at sensing blood sugar and releasing the measured amount of insulin. The higher the blood sugar, the more insulin that will be released. And the other thing is it's good at shutting that off too, because hypoglycemia, low blood sugar, is a big problem.
Dangerous.
It is. It's very dangerous, and especially a problem for type 1 diabetes. So
when you implant the device, how big is it to begin with?
Right. So you're right that we're implanting the device, and inside it are the cells, so it's a combination product. This isn't a device therapy, it's really a drug therapy. We're regulated as a biologic. And that product is about the size of a credit card, we'd like to say, or a business card.
Okay.
So it's about that shape, almost that thin, so it's about 1 millimeter thick. and the current device is 3 by 8 centimeters. And right now, that's a device configuration that allows us to do dose escalation in this first trial. So patients are getting different numbers of devices, and the number of products that we implant over time in the trial will increase, allowing us to control the dose.
How do you control the dose once it's already been implanted into the patient?
Yeah, it's almost a misnomer dose. It's sort of a capacity for dose, because the cells themselves are providing the insulin in a physiological sensitive manner, right? So the dosing really ends up being done by the beta cells themselves. We provide a capacity for that dose by providing a certain number of cells in a certain number of devices.
So as I understand it, when you and I eat, not being a diabetic, there are K cells roaming around in our body that pick up elevated blood sugar levels, and they release a peptide that is sent back to the pancreas, instructing it to produce insulin. Is your device in essence going to be doing the same thing? Or how, if not that way, then how does it read elevated blood sugar levels?
So the K cells you mentioned are in the gut, and they also sense digestion and taking in a meal, and they'll release a hormone called GLP-1 that potentiates the action of the beta cells in the pancreas to release their insulin. Beta cells also have a direct ability to sense that elevated blood sugar even outside of that K cell input. So they can sense blood sugars directly and release insulin that way. Obviously, the physiology is very complex, and we don't fully understand it at this point, but suffice to say the beta cells communicate well with the host. And this is data that we can speak to from preclinical models, and we hope it translates well over to human, but that it would interact with the patient just like their normal beta cells in the pancreas, even though they're not in the pancreas, they're under the skin and inside a device.
There's a final break. If you can hang on for just one more moment, it's exciting to wrap this up. We'll be right back. Conversations That Matter is a not-for-profit program made possible thanks to the charitable support of the following and from viewers like you. Please visit conversationsthatmatter.tv and help us to continue to produce this program. Thank you. Holy smokes, I'm just trying to think about how you work your way through to make that work. So you implant the device. What's the life of it once implanted into the patient?
We don't know that yet, so that's one of the objectives of this very first trial, is to see how long the product will last. So in our preclinical models, which have been rodents with no immune systems, they only live a year, and we've seen that the product can function out through the year, the lifespan of those preclinical models. So one would think it would last a year in a human, probably may even last longer, but we can't test how long it will last until this is one of the points of the clinical study, until we run clinical studies to see when it comes on to function and how long it will last.
So then that means that this is a treatment. It's not a cure, but as far as treatment is concerned, it's probably the closest to mimicking the natural process the body goes through.
Yeah, it is a treatment, and if it works in people like mice, it could actually become what we would consider to be a functional cure. So we haven't changed the autoimmunity of the disease. We've replaced the cell that's lost to autoimmunity and protected it from that autoimmunity.
Where are we at now that we go to phase 2 of human trials? That's coming up.
We're in a phase 1, phase 2 trial right now. So this sort of serves both purposes. This first cohort, which we're still active in right now, you could think of this as the phase 1 trial part. It's evaluating primarily safety and tolerability of the product. Once we've established that well and made sure that the product has biological function, so to speak, we would then be looking to increase the dose in a second cohort. In that second cohort of this trial serves early phase 2 trial purposes, in that it's also looking to evaluate early efficacy of the product.
You know that diabetics are thinking, okay, when will this happen?
Right. It's really hard to make a projection like that because
You know, that's not the answer that they want.
there are unknowns
Yeah.
to all trials. If things went well as planned, you could be talking, this first trial is a 2-year implant duration. So we could think of it in terms of a trial that lasts about that long, so at least another year or so, triggering the next trial, which it might be the pivotal trial that would allow approval of a product like this. So, of course, that's a trial that needs to be larger in scope. You're talking probably
A decade.
I don't think it's that long.
Yeah.
Three,
But
four,
could be.
3-4 years for a trial like that, a pivotal trial, perhaps. But that's why I'm being, I guess, cagey.
Well, I know you
know,
have to
but-
be careful because
because I don't want to set a false expectation. That hurts people in the long run.
Right. But this is one of the most encouraging things that I've seen as far as being able to deliver a treatment that can free diabetics from the day-to-day process of pricking their fingers or having a pump or whatever that's monitoring their glucose and not having to carry something around externally. It would be incredibly liberating.
Yeah, it would. And that's what actually motivates me and all of us at ViaCyte is that what we're working on can really change people's lives, in the end. And it's immensely motivating to come in and do the hard work day after day. And, science is kind of like a bunch of baby steps. And sometimes you're going flat for a while and then you get a breakthrough and then it's flat for a while. But, it's part of the gratification to be able to look back, like, myself over a decade's worth of work and say, wow, we have been making steady progress that's now reached the clinic. It's really moving.
You talk about that gratification. What was that one moment in this journey that you look back on and go, oh my gosh, I actually think we can do this?
It was the approval from the FDA, or the allowance to start our clinical trial. Literally, tears came to my eyes when we got that letter from the FDA because what we were doing was really innovative, and others had trouble getting their stem cell-based therapies into the clinic, and we just didn't know if we were going to be allowed to start yet or not. What we did know is we'd pretty much run the course of what we could learn in the animal models, and it was high time to start, translating this into patients. So that was the most gratifying moment I've had so far in the 13 years at the company.
So when did you get that letter? Do you remember where you were when you got that?
Yeah, August 14th, 2014, so it was a really great moment a little over a year ago.
Yeah. Time gets measured as in before and after from then on.
Yeah.
Well, I wish you all the best, and
Thank you,
so
Stuart.
much for coming in and explaining this to us because, as I say, it's very encouraging for people who are dealing with type 1 diabetes.
Thank you.
Thank you.